Proteins are extremely important macromolecules in living organisms. More than 50% of dry mass in the cells is proteins. Proteins has many important functions, for example, to produce hormones, to produce antibodies, to support cells, to replace dead cells, to carry oxygens in the circulatory systems. Despite their tremendous range of functions, all proteins are made from the same basic subunits or monomers, which we call as amino acids. Proteins is a long-chain amino acids linked together by peptide bond. All amino acids are compound with similar basic structure. They have amino group, a carboxyl group, and a side chain or R group that is specific to each amino acid. There are 20 types of different amino acids. All the different proteins are made from these 20 amino acids. The variations in the proteins are caused by the order of the amino acids, the chain length, and the folding in the chains in a distinct ways. How proteins form? Look at this picture. This picture shows how two amino acids are joined together. One loses the hydroxyl group from its carboxylic acids group, while the other loses the hydrogens from its amine group. This leaves the carbon from the first amino acids are free to bind with the nitrogen's atom from the second. This link we call as peptide bond. So the oxygens and the two hydrogens atom will leave the amino acid to form the water molecule. There are four types of protein structures, which are primary structure, secondary structure, tertiary structures, and quaternary structure. Let's talk about the primary structure. Primary structures is the linear sequence of amino acids. A polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. And we know the primary structure is the linear sequence of amino acids. The amino acids in the proteins are joined by peptide bonds, which link the amino groups of one amino acid to carboxyl group of another. A water molecule is released each time a bond is formed. Next, secondary structure. Secondary structures is the structure of protein molecule resulting from the regular coupling or folding of the chain of amino acids. This protein chain often falls into two types of secondary structures, alpha helix and beta sheet. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. Next, tertiary structures. Tertiary structures is the complex structures of the protein molecule resulting from the three-dimensional of the coiling of the already folded amino acid chains. The tertiary structure of a protein is three-dimensional shape of protein chain. The shape is determined by the characteristics of the amino acids making up the chain. There are two types of the tertiary structures protein, which are globular proteins and fibrous proteins. A protein whose molecules curls up into the shape of ball, such as the myoglobin or hemoglobin, is known as the globular proteins. Many other proteins do not curl up into a ball, but they form a long strange. These are known as the fibrous proteins. Now, let's talk about the difference between globular proteins and fibrous proteins. First, the shape of globular proteins is spherical, while the fibrous proteins is elongated string. Second, globular proteins is soluble in water, acid, and bases, while the fibrous proteins is insoluble in water, weak acid, and weak bases, while it's soluble in the strong acid and strong bases. The third one, globular proteins, has a weak intermolecular hydrogen bonds, while the fibrous proteins has a strong intermolecular hydrogen bond. The example of globular proteins are hemoglobin, insulin, caseins, and many more. While the example of fibrous proteins are elasins, keratin, and silt. The last one, quaternary structure. Quaternary structures is the three-dimensional arrangements of two or more polypeptides. Two or more polypeptide chains can come together to form one functional molecule with several subunits. For example, the four subunits of hemoglobin cooperate so that the complex can pick up more oxygen in the lungs and release it in the body. Nucleic acid. Nucleic acids is a group of biomolecules that is found in all living organisms, and they use to store the genetic information. Nucleic acid is also used to produce the proteins. Nucleic acid made of the monomers, which we call it as nucleotides. Nucleotides has three components, which are the 5 carbon sugars, or we call it as ribose, a phosphate functional group, and a nitrogenous base. A DNA, the 5 carbon sugar is deoxyribose, while for the 4 nitrogenous base are adenine, guanine, cytosines, and thymine. DNA is composed of the strands that is arranged as right-handed helix. So these strands are antiparallel, meaning that they are orientated to the different directions. So these two strands are held together by the hydrogen bonds. So the cytosines will form hydrogen bonds with the guanine, while the adenine form hydrogen bonds with the thymine. So this complementary pairing of bases will link the two strands together, and this plays an important functions in the replications of DNA molecule. Besides DNA, there are the other types of nucleic acid, which is RNA. RNA differ from DNA because first they contain the ribose sugar instead of deoxyribose. Second, they contain a nitrogenous base uracil instead of thymine. RNA usually is single-stranded and it doesn't form a helix. Because of this, they are less stable and they are temporary. So DNA are used to store the information to produce the proteins, while RNA functions in regulating these informations in the protein synthesis process. I think that's all about our lesson today. Don't forget to do the exercise on Edmodo, and see you!